Hi, everyone. My name is Avantika, and today I'm going to talk about oculomics, oculometrics, and neurological disease. Can you see my screen? Can you still see it? Sorry. Yeah, so it's blurry, right?
Yes.
So blurry vision or double vision is a huge concern. Each year, over 100 million eye exams are performed for this reason. And one of the main causes is that it can be linked to functional causes of how the brain is processing visual information. This can lead to a lot of these diseases out here, but they require the expertise of a neuro-ophthalmologist to help diagnose. With only 700 of these specialists in the US, you can imagine how challenging that is. Today, I'm going to tell you how oculometrics can be a game changer in bringing that expertise of neuro-ophthalmology down to the level of primary and secondary care. I'll focus on these two conditions and tell you about our data that we have so far. Now, oculometrics, or the science of eye movements, can be used in an objective and quantitative way to increase healthcare provider efficiency, reduce clinical error, as well as improve a patient's quality of life. The way we are doing this is through our device called NEOS, which integrates a head-mounted VR display with interactive gamified stimuli that elicit specific eye movement responses. These are captured by high-frequency infrared cameras and then processed by a customized software platform to produce easy-to-read, quantitative oculometric reports that capture different aspects of afferent and efferent function in under 12 minutes. So how does this play a role with Parkinson's disease? Unfortunately, Parkinson's is a very difficult condition to diagnose and can take over 2 years. There's no single diagnostic test, and that is why there's a need for objective biomarkers. This is where eye movements can be a game changer, and Professor Konrad Weber is actually leading a study right now at the University of Zurich, as well as the University of Exeter, where they're developing AI/ML models to facilitate diagnosis as well as monitoring of individuals with Parkinson's using oculometric and pupil function biomarkers. These are also being correlated with current disease stage as well as health economics data. To date, we have collected data on over 258 individuals, 103 of whom have Parkinson's disease, and 7 individuals have atypical Parkinson's. All participants undergo 3 exams, which involves a manual clinical exam, a device measurement with our device, as well as gait analysis. All groups are then compared with where age and sex matched, as well as we're then using supervised machine learning algorithms, so no black box algorithms out here, to then help distinguish Parkinson's versus healthy control data based on multimodal feature-based extraction on most relevant oculometric parameters. To give you an example, out here you can see in clinic, one way to distinguish healthy individuals from people with Parkinson's is looking at their vertical saccades. For a healthy person, they can move their eyes up and down, but an individual with Parkinson's will have difficulty. We took this subjective clinical test, objectified it in a standardized way, and then charted that oculometric data. Out here you can see for a healthy person, how that data would look in terms of latency and accuracy, and then we normalize that data to show you that if you're in that normal orange zone, you're healthy. However, for a person with Parkinson's, this is not the case. You see that their data is much more different, as well as it's not in that normative zone that you would like to see. Now, our models are still being processed, but we are very happy with the preliminary data results that we have so far in terms of sensitivity and specificity for both our cohorts. We have a Breyer score of 0.06 as 0.13, where 0 is seen as perfect. So we're pretty happy with that. Now, how does this play a role with multiple sclerosis? Again, MS is a very challenging condition, and it's hard to diagnose. This is where Dr. Anke Salmon, who's a neurologist at the Ruhr University in Bochum, Germany, believes that functional eye movement biomarkers could be a great way to help in distinguishing typical versus atypical features. We've been working with her group, and one of the things that we're focusing on is how can we help improve with optic neuritis identification. This was included last year in the diagnostic criteria of MS, but to identify optic neuritis, you need to see if there is indeed a positive relative afferent pupillary defect identified using the swinging flashlight test, which is unfortunately riddled with a lot of clinical error. So we took this test and then we digitized it, and we came up with the simple score. If you're in that orange zone, you're healthy. But But if you fall on either side, that means you have a relative afferent pupillary defect. Currently, we are validating this score in a group of over 759 individuals, 397 of whom have MS, but we also have individuals with other neurological conditions, from demyelinating disorders all the way to neuromuscular disorders as well. All individuals undergo 2 independent functional tests by a neurologist, including the swinging flashlight exam and an exam with our device, and some of them also have some OCT data being collected. We're very happy to say that we have good clinical sensitivity with our test, with an AUC value of 0.79. Additionally, what we found that in a subset of people with OCT findings, not all of them had a positive, not all of the ones with a positive APD had structural changes, meaning that sometimes functional changes are preceding structural changes, making it a good clinical biomarker. We're also now working to detect intranuclear ophthalmoplegia, which is a horizontal gaze palsy, and bilateral INO is most commonly associated with MS. To date, what we have done is we've collected data focusing on horizontal saccades, and just finishing up.
Thank you.
And so far, we've seen some very positive findings. Do come and check us out at our ARVO with our poster. And with that, I just want to say that not only in the clinic, but also in drug development, these biomarkers can be used for patient identification, endpoint quantification, as well as safety monitoring.